This is Biology 223 Genetics, and in Russell's chapter, Chromosomal Basis of Inheritance, it's either chapter 10 or 11, depending on the edition. There's a problem 19, let me read it. The horse has a diploid set of 64 chromosomes. So the horse is Equus calibus. Know this is a horse, 64 chromosomes. And the donkey, that is Equus asinus, has a diploid set of 62 chromosomes. Mules are the viable but usually sterile progeny of a mating between a male donkey and a female horse. So the donkey comes as a jackass. Ass is the word for a donkey, as you see. And the female is a jenny. And in the horse, Equus calibus, the male is a stallion, and the female is a mare. Mules, so we cross typically here, so we cross a jackass with a mare, and we get a mule. Mules are the viable, usually sterile progeny of a mating between a male donkey and a female horse. How many chromosomes will the mule cell contain? So I want to expand this problem because there's many issues here that come up. The question itself is asking a relatively obvious one. So let's look at this. So we have 64 chromosomes, so we have 32 in Equus. So Equus calibus, we have 32 pairs, or we have, if you think of that, we have 31 pairs of autosomes and XY. Their placental mammals, sex determination is XY. In the same genus, the donkey is 62 chromosomes, so it has, let's say, 31 pairs, of which 30 are autosomes and XY. Okay, so this is a classic situation where you say, well, of course, the mule here, that poor mule then, must have 31. So the mule, the gamete from calibus is going to have 32 chromosomes, and the gamete from the asinus is going to have 31 chromosomes. So the mule is going to have 32 plus 31. That's going to be equal to 63. So when it undergoes meiosis, there's a problem of dividing 63, an odd number, into 2, and that means that there's likely to be non-disjunction. Okay, we'll talk about that a little bit later here. The other thing that may be relevant is they're separated common ancestors about 4 million years. And you might wonder, how does speciation occur to cause different number of chromosomes? What happened where a chromosome, presumably one of them has 2 short chromosomes. I'm going to draw them in this way to think about another point. One of them has 2 chromosomes, the other has 1 chromosome. Let's be the simplest sort of thing. And so the horse has 2 chromosomes, where the donkey has 1, and presumably all the genetic information is the same. This is broken up, and whether the common ancestor had 1 or 2 and 2 chromosomes fused, or 1 chromosome split in 2, I don't know. But how that might happen is rather puzzling, and one might think about it. Those are other examples where species differ, and apparently within zebra species there are quite a few differences. All the members of the zebras have different numbers of chromosomes, so that's a puzzle. But I draw it this way to illustrate that. So we expect that the homologous chromosomes from, let's say here, Equus calibus and Equus asinus are going to align, and there's going to be some sort of crossing over to align them during prophase of meiosis 1 in the mule, right? The mule undergoing gametogenesis is going to duplicate DNA to make these bivalents. They're going to associate through crossing over and at strand invasion, all that. And at the metaphase plate, we'll see clearly when they're lined up. And then during anaphase, they're going to be pulled apart by the spindle apparatus. And now we see the problem. We have the problem that there's an odd number here, so the crossing over might cause complications. And even then, in meiosis 2, during anaphase, even if they manage to somehow line up in some funny way, you can imagine, think through that, and you will realize that the likelihood is we have a non-disjunction. And non-disjunction means that the gamete progeny will come up, and with the gamete from the other member mating, there's a likelihood of an extra chromosome or an absence of a chromosome. Well, an absence of a chromosome, we're not surprised if that's lethal. There may be missing genetic information. An extra chromosome, so we expect embryonic lethality. In the case of an extra chromosome, well, we know obviously the case of trisomy in humans, Down syndrome, in which an extra chromosome, it's not lethal, but it has gross morphological effects. And you might wonder, well, why do we only have some forms of trisomy observed in humans, and what about all the other chromosomes? And presumably they're embryonic lethal. So non-disjunction, that is, which results in having an extra chromosome or a missing chromosome, is likely to be lethal, and so the mule is not going to have, it's not going to be fertile. Let's examine this problem a little more. So let's consider the mule. So I'm going to divide it up this way: a male mule and a female mule, and a male hinny. What's a hinny? A hinny, am I spelling this right? I think that's with an E there. And a male hinny, a female hinny. A hinny is when a stallion mates with a jenny. I'm not sure about that E there. And let's go through this. So what's interesting is mules look different than hinnies. Okay, sure. One has a horse mother and a donkey father, and the other is the other way around. So so that's a way to do it. So let's look at autosomes here and sex chromosomes, and let's consider Equus calibus, horse, Equus asinus, Equus calibus, Equus asinus. So if we look at the male mule, we say, okay, well, it has 31 autosomes from no, I think I have this wrong here. Sorry, the mule is going to have 31 from calibus and 30 from asinus. And likewise, the female also, mule, is going to have the same. And the hinny, of course, is going to have the same. So genetically, autosomally, they all are the same. So why do they look different? Well, let's look at the sex chromosome. The mule comes from a jackass father, so it's going to have an Y chromosome from asinus and an X from the horse, while the male hinny, the other way around, the stallion with the jenny, it's going to have a Y chromosome from calibus and an X chromosome from asinus. So you could say, well, the reason why a male mule and a male hinny look different is one has an X chromosome from the horse and the other from the donkey, and the Y chromosome vice versa, and maybe that's to blame for it. But the female mule and the female hinny look different, and you see that they are then genetically identical. So what could be responsible for that? Is it that one has a donkey mother, So this is the question then that becomes interesting. What genetic similarity? What's the difference? Is it that the hinny the hinny foal drinks donkey milk, and the mule foal drinks horse milk? That's not likely to seem very important. Is it the gestation is different in a donkey or a horse? So I don't think we're going to assume there's very likely that the milk or the process of gestation is going to be very likely. So what about the oocyte? So the oocyte, now we have, this relates to, so I want to point out that this problem relates to other issues. So this is what is from the chromosomal basis of inheritance, and also you're going to want at the chapter on extensions and deviations from Mendelian Genetic Principles. That's chapter 13, and there are problems there. And you might think about that. Well, what about the oocyte factors? The oocyte is going to contribute all sorts of factors, including the mitochondria. Well, we don't expect gross morphological differences because of mitochondria. That just doesn't make any sense. Larger ears looking like a donkey rather than a horse, vice versa. That doesn't seem very likely. All sorts of oocyte factors. We can imagine there's a vast amount of factors in an oocyte that are contributed. And so the volume and the early embryo, contribution to the early embryo and later development of factors that come in the oocyte are far more than that of the sperm. So could it be those factors from the oocyte versus the sperm? I can tell you there's some complexities here, especially with regard to the sperm contribution, which are beyond the scope of the book. We'll see in a research article I'll talk about later in a different video. And then the other aspect that should be looked about is gene regulation. So there's regulation of gene expression in eukaryotes. And that chapter, they talk about imprinting, genomic imprinting. And that's where the methylation patterns, which we've talked about before, there are other mechanisms by which chromatin can be affected, but let's just consider the simple one. So if we look at cytosine here, we can see that at the 5 position there can be a methyl group, and whether this methyl group, let me draw it here, is there or not, causes genes to be expressed. There are these CPG islands. I've spoken about this. So we have CPG islands in promoters, and some of this can persist. So whether or not an organism, including humans, inherits a specific allele from the father or the mother affect development. So we have imprinting, genomic imprinting, and how that happens is related also not just then to factors from the oocyte, but the oocyte and the sperm DNA have methylation patterns that are laid down during gametogenesis, and they have long-term consequences and a very complex process of development, beginning with zygote formation, and in a lot of it's in early embryogenesis. So that's to think about this, and to think about all the other dimensions of biology that occur in this. So we have some examples of some almost trivial problems giving us a window into many complex aspects of biology and genetics. I will end